Have you ever wondered if a part of you is, in a way, immortal? In this explainer, we're going to dive into the incredible story of the cells that carry your legacy forward. It's a tale of biological eternity, of some really precise engineering, and two reproductive strategies that operate on wildly different clocks.
So let's just jump right in with that provocative question. We think of our bodies as temporary, right? Destined to age and, well, pass on. But what if a part of you, a really specific lineage of cells, is, in a sense, designed to live forever through your descendants?
To get to the answer, we need to understand this fundamental split that exists inside your body. A division between the cells that make you you today and the cells that are literally destined to create the future.
Okay, so your body is basically built from two types of cells. On one hand, you've got your somatic cells. These are the workhorses building your skin, your heart, your brain. They do their job, but their story ends with you. But then you have the germline cells. Think of them as cellular royalty, the ones carrying the blueprint for the next generation.
And this germline, this is the whole story right here. It's not just any group of cells. It's a direct, unbroken chain that stretches back through countless generations. This is the biological thread that connects you to every single ancestor you've ever had. And it has the potential to carry your legacy forward, well, forever.
So how does a germline cell get ready to create an entirely new person? It goes through this highly specialized process called meiosis. And this isn't just simple cell copying, oh no. It's an elegant multi-stage dance designed to carefully and precisely have the genetic code.
You know, its main job is to do one thing with absolute precision. Take what we call a diploid cell, which has two full sets of chromosomes, and create haploid cells, which have just a single set. And this step, it's completely non-negotiable. It makes sure that when a sperm and egg combine, the new embryo has exactly the right amount of genetic material, not too much and not too low.
All right, so let's take a closer look at how this all works. The next few slides are going to walk us through the key stages of this really elegant cellular dance.
It all kicks off in what's called prophase 1. Here you can see the homologous chromosomes. You get one from your mother, one from your father, and they're pairing up. And then something pretty incredible happens. See that point labeled crossing over? They are literally swapping pieces of genetic information. This is what shuffles the genetic deck, and it's what guarantees that every single sperm or egg is totally unique.
Next up, in metaphase 1, those pairs get organized. They don't just float around randomly. No way. They align perfectly down the center of the cell. And this precise arrangement is so crucial because it sets the stage for the first big split.
And in anaphase I, that split happens. The pairs are pulled apart, with one full chromosome from each pair migrating to opposite ends of the cell. This is it. This is the exact moment the chromosome count is literally cut in half.
So what you're seeing here is the grand finale. The two new cells, they immediately jump into a second division. In metaphase 2, the chromosomes line up again, and then in anaphase 2, their individual strands, the sister chromatids, finally pulled apart. This leaves us with the final product, 4 genetically unique haploid cells, and each one is ready for its potential role in fertilization.
Now, here's where it gets really interesting. While those basic steps of meiosis are universal, the way they're applied in creating sperm versus creating an egg, well, it's dramatically different. It's like the same fundamental process, but it's run on two completely separate operating systems. Right here, you can see the two paths laid out side by side. On the left, that's spermatogenesis, basically the sperm factory, and on the right, you've got oogenesis, the process of making an egg. The visual difference is, well, it's stark, right? The male process results in 4 identical-looking sperm, while the female process focuses all of its resources on creating one single massive egg and a few tiny little polar bodies. Let's break down these key differences, because this is where our story really takes a fascinating turn. I mean, just look at the timing. For males, this process kicks off at puberty and just runs continuously for life. For females, it starts before birth and then just stops, hits the pause button. The duration is also staggering. A few weeks for a sperm cell, but it could be up to 50 years for a single egg to complete its journey. And you know, these differences in timing, they're not just some random biological trivia. They are a core feature of human reproduction, and they create this profound biological asymmetry that has really significant consequences.
So let's actually trace these two timelines, because it's pretty wild. In males, the germ cells are just dormant until puberty, and then boom, they become a non-stop factory, churning out millions of sperm. Now, in females, all the potential eggs a person will ever have are already formed when they are a fetus, beginning that first stage of meiosis. Then the whole process is arrested, paused, for 10, 20, heck, even 50 years, until one cell gets chosen to mature each cycle.
So, why? Why does this super long pause in the female timeline matter so much? What are the risks of putting such a delicate cellular process on hold for decades? This is really the crucial point. Well, it turns out that hitting pause on this delicate cellular machinery for so long, it comes with risks. When the process finally restarts, sometimes decades later, the components that are responsible for separating the chromosomes can become less reliable. And this increases the chance of errors, which can lead to an egg with the wrong number of chromosomes, a condition that we call aneuploidy. And look, these separation errors, they aren't just some abstract idea. They can have very real consequences for development. An embryo with an irregular number of chromosomes often won't develop, but some, like those with an extra copy of chromosome 21, can result in conditions like Down syndrome. It really shows you how this tale of two timelines is absolutely central to human health. And that leaves us with this final thought to just kind of chew on. You've got one strategy, continuous, mass production, and the other, a long-term paused investment. This fundamental difference in how we create the cells that create the next generation has been shaped by millions of years of evolution. So what does this deep, deep asymmetry really tell us about the very different strategies of life itself?